Hey everyone, and welcome back. Today, we're diving into a technology that is just mind-blowing. Imagine giving a microphone to every single cell in your body. It's called single-cell RNA sequencing, and it's letting us hear the individual stories that, until now, were totally lost in the crowd.
So think about this for a second. How do you listen to the whisper of one single cell when it's surrounded by the roar of millions of others? How do we go from this blurry, averaged-out picture to seeing the unique genetic fingerprint of each and every cell? Well, that is exactly the promise of this technology.
Okay, to really wrap our heads around this, let's use an analogy. Picture a massive, bustling city. But instead of people and buildings, this city is a complex tissue in your body, like your brain or maybe even a tumor. And every single cell is a citizen, with its own job, its own story, and its own part to play. Now, for the longest time, the way we looked at this cellular city was, well, kind of blurry. Traditional methods were like reading the city's newspaper. Sure, you get the big headlines, the averaged-out news of the day. But with single-cell sequencing, now we can eavesdrop on individual conversations happening on every street corner. We get to hear the whispers, the secrets, every unique story. It's a total game changer. So this brings us to our first big idea, the microscopic gold rush. This is all about that incredible leap in technology that took us from seeing a fuzzy, blurry average to a super sharp, high-definition view of every cell.
So, how on earth did scientists crack this code? How did they figure out how to pluck one tiny single cell from a sea of millions and then do it again and again and again with just mind-boggling precision?
Well, this was the first giant hurdle they had to overcome. Using these incredible technologies like microfluidics, and you can just think of those as tiny microscopic plumbing systems that can handle individual cells inside of tiny droplets, scientists finally found a way to isolate them, one by one. And that single breakthrough, it was the key that unlocked everything else. And you know, this quote just nails it. It's like microscopic fishing for biological gold. It really gives you a sense of just how tricky this is, and how precious every single piece of data we get from a cell truly is. Okay, so we've caught our little piece of biological gold. What's next? Well, now we have to read its diary. And that brings us to our next section, where we're going to dig into how scientists decipher the transcriptome, which is basically just the complete list of genes that are active, or switched on, in that one cell at that one moment. Because, catching the cell is just the start. The real challenge is, how do you actually read its genetic diary? How do you peek inside and see which genes are firing on all cylinders and which ones are just taking a nap? The answer is this absolutely brilliant idea called cellular barcoding. It's so clever. Scientists figured out a way to attach a unique molecular tag, basically a tiny piece of DNA that acts like a barcode, to all the genetic messages, the mRNA, from a single cell. So every cell gets its own personal, unique barcode. A really good way to think about it is like giving a personalized library card to every single cell. That way, even if we take all the books, or in this case, all the genetic messages from the entire city's library and throw them into one big pile, we can still trace every single one back to the person, or cell, who checked it out. So the process basically looks like this. First, you grab all the active genetic material, the mRNA, from each of your isolated cells. Step 2, you stick that unique barcode onto it. And then for step 3, and this is the really efficient part, you can just pool everything together and sequence millions of these messages all at once, because you know you can sort them out later using those barcodes. It's just incredibly smart. All right, now for the real magic. We've got the data, but how do we turn that raw data into an actual discovery? This is the part where we get to unmask the true identity of every single cell. I mean, it's one thing to collect millions of these barcoded gene sequences, but it's a whole other challenge to make sense of it all. How do you find the real biological story that's hidden inside this absolute mountain of data? And that's where the computers come in. We're talking powerful computers and really sophisticated software that act like master detectives. They sift through the millions of conversations we've recorded, and they look for patterns. They start grouping cells together that are talking about the same things. It's kind of like identifying the different neighborhoods and communities within our cellular city. And this is the payoff. All of that incredibly complex molecular biology, all that heavy data science, it's all about one simple goal: turning the complete chaos of millions of individual cellular actions into a crystal clear picture of what's actually going on. So what do these patterns actually show us? Well, using these amazing visualization tools, you might hear names like UMAP and TSNE, we can create a literal map of our cellular city. This lets us spot the really rare and unusual things, like, say, finding a handful of rogue cancer cells hiding in a tumor. We can even watch a newborn stem cell as it grows up and decides to become, say, a neuron. It's like having a window into the most fundamental processes of life itself. So let's bring this all home and talk about why this is such a huge deal. Because this isn't just some cool science experiment in a lab. This is the future of precision medicine, and it changes everything.
And I want to be really clear. This is not science fiction. This technology is being used right now, today, and it is fundamentally changing how we approach everything, from basic biology to treating patients in the clinic. The impact is happening as we speak.
And here are just a few real-world examples. Scientists are using this to create personalized cancer therapies that go after the specific cells driving a tumor. They're using it to understand what goes wrong, cell by cell, in diseases like Alzheimer's. And they're using it to map out the insane complexity of our own immune system.
Ultimately, this is where we're all headed. The big dream is a future where treatments aren't just for a general disease like cancer, but are tailored to the precise molecular signature of the exact cells causing the problem in one specific patient. That is what true precision medicine looks like.
And that leaves us with one last fascinating question to chew on. Everything we've talked about today was about RNA, the cell's active messages. But what happens when we can combine that information with a cell's DNA, its permanent blueprint, and its proteins, its little workers, all at the same time from the same single cell? What mysteries are we going to solve when we can finally paint a complete, multi-layered portrait of every single cell? The possibilities are just endless.